from this, we can see very clearly that crystal lattice contacts have got absolutely no impact whatever on the observed conformations of alpha and beta subunits. So any conclusions that we draw from those structures are not influenced by artifactual considerations. And as I'll show you in a minute, they help to define the chemistry of the catalytic cycle of how ATP is made and hydrolyzed. Now, with the position of the central stalk, it's a different story. So what we found there is that although we could see rotational displacements of the foot of the central stalk in different pictures, there was actually no correlation of that rotation with biophysical observations that I showed you just a few of those a moment ago. And the position and the rotation of the central part is actually dependent upon lattice contacts. But what this comparison has shown us is something extremely interesting that was suspected already. And that is that this rotor in the middle, which I've not pointed this out, is an alpha helical structure, actually has points of weakness in them. And that's shown here. So what we've plotted so these are the alpha helical regions. This is actually the N-terminal part of the protein. This is the C-terminal part, and they make a coiled coil in this region here, and this constitutes the rotor that's turning around inside the ATPase. And what we can see by looking at the crystallographic data is that these points, these regions colored here in yellow and green and red, are flexible. And that's a very interesting observation, because about 10 years ago, we did a molecular dynamics simulation with Martin Karplus to look at what happened to this structure in a 120-degree step. And the conclusion from the MD simulation was that in the 120-degree step, the helices came apart. Of course, they're together in the ground state that we observe by crystallography. But they come apart, and then as you go through the step, the contacts and the structure reforms. So we think this is part of this point that I made, that although the ring is rotating continuously, this part is stepping, and there is storage of energy in this region here, which is released in a quantized fashion to give the stepping motion that was observed. And this shows that it's no better when you solve structures of the catalytic domain with the ring attached. The ring gets pushed aside in the gray structure relative to the brown one. So the brown one is the bovine F1C ring structure, and the gray one is the yeast F1C ring structure. And in the case of the yeast enzyme, the ring has been displaced, and that, as I've just pointed out, is because of flexibility in this region here. So what do we know about the mechanism of ATP hydrolysis? Well, what I've shown here is one of the catalytic sites of the enzyme, and I've shown what happens to it as these 120-degree rotational steps take place. So we start out here with a state that's empty. So this is Paul's open state when no nucleotide is bound. And the reason it's not bound is there's no nucleotide binding pocket. So in order to form a nucleotide binding pocket, these two aromatic residues have to come close to each other and be parallel to each other, and then an adenine ribose can bind in this pocket. So that's what happens in the first part of the catalytic cycle. As part of the closure of the open state, those two residues come close together, and then ATP can bind, as shown here, in this state of the enzyme. And then in another site, the ATP is released, and then this site, so this is the loose site, the loose state. When the ATP is released from another site, this loose state converts to the tight state, and this is the catalytically active state where all the catalytic residues are in place to bring about the synthesis or the hydrolysis of ATP. And then if we think about ATP hydrolysis, the transition state forms, and we observe this by forming transition state analogues. And then, of course, eventually this bond is cleaved, is hydrolyzed, phosphate is produced and is released. But what was not known until recently was actually the order of release of substrates. So we now know that phosphate is released first, and then this structure, which is still not published, the magnesium has been released, but the nucleotide is bound. So the order of release of products, sorry, not substrate, products, is phosphate, magnesium, and finally the nucleotide, which then regenerates this open state here, where no nucleotide is bound. And what we found in these structures we've determined recently is that if we compare the structure in the region where the magnesium is bound in the active state of the enzyme where catalysis has just occurred or is about to occur, the magnesium is coordinated and held in position here by this hydroxyl, which is on this threonine residue, which is part of the P-loop sequence or the A motif sequence, and by this phosphate. And then there are water molecules that are held in place here, which provide the other coordinating ligands. Now, what happens then as the rotor turns is that the nucleotide is moved, still bound, is moved to the right as the site opens. This destroys the water sphere and the coordination of the magnesium, and it then just drops out of the catalytic site, leaving the nucleotide bound. And then in the final step, as the opening is completed, the nucleotide falls out. So we've been able to resolve all of these steps now by X-ray crystallography. So I want to turn now to how the enzymes are regulated. And I've said that the mechanisms of regulation differ between different kinds. So, but many of them are regulated by ADP inhibition. So if you hydrolyze a lot of ATP, the accumulating ADP will actually slow down and inhibit the enzyme. That's common to many of the enzymes. The mitochondria have an additional mechanism for preventing the enzyme from hydrolyzing ATP when proton motive force has been dissipated, for example, during ischemia. And they bind a regulatory protein called IF1, inhibitor of F1 ATPase. This protein is not found in bacteria or in chloroplasts. And there it's been suggested that one of the proteins that forms part of the rotor assumes the role of the inhibitor protein. So this is the epsilon subunit, and I'll show you that. And very recently it's been realized by Mexican workers that in some bacteria, exemplified by Paracoccus, there is actually an inhibitor protein, given this Greek letter, but it's not related to the mitochondrial inhibitor protein. Not much is known about this. Another very important point to be made is that although the mitochondrial enzyme can operate either in either direction to make ATP or to hydrolyze it until it's inhibited by this protein, not all bacterial ATPases can actually hydrolyze ATP. So you find when you isolate them, they are latent. And the mechanism of latency, inhibition, is not understood. And then another mechanism of regulation obtains in chloroplasts. So in chloroplasts, during the hours of darkness, unless there were a mechanism to hydrolyze the ATP synthase, it would simply hydrolyze all the ATP that had been made. To prevent that wastage, there's a mechanism where a disulfide bond forms in the gamma subunit, controlled by other proteins. So what is happening is that the redox condition of the chloroplast is being sampled by these proteins and feeds back to the ATPase to inhibit it. When daylight returns, reducing conditions are re-established. The disulfide bond breaks, and the enzyme can then make ATP. So this is what the mitochondrial inhibitor protein looks like. It's a very simple alpha helical protein, and its C-terminal regions form an antiparallel coiled coil. The inhibitory part is at the N-terminal end, and you can actually dispense in the laboratory with this part here, and the remaining part of it is still a very effective inhibitor protein and easier to study from a structural point of view. And this is the way it works. It goes in to a catalytic, so that's a catalytic interface, and stops the enzyme dead, rather like putting a stick into a bicycle wheel. Now, the entry of the protein into that site requires the hydrolysis of 2 ATP molecules to close the site and entrap the protein. There's a tunnel which leads upwards to the catalytic site from the membrane region. So the membrane would be here. There's the tunnel. And it forms interactions with the catalytic protein, which is the yellow one, and also with the rotor, and stops the enzyme from turning. We've studied the details of the interaction of the inhibitor to find out what are the important residues by changing every one of them individually to alanine and then measuring K-on and K-off. And almost all of the binding energy comes from hydrophobic interactions. Now, recently what we did was to do this. So initially we solved this inhibited complex using the bovine enzyme, where the inhibitor is in brown. And recently we solved the yeast enzyme with its inhibitor bound, very similar in sequence. We found it bound in a similar position, but they didn't bind in exactly the same way. And you can see that here. So here is the rotor. So those are those 2 helices again in the rotor. The N-terminal parts of both inhibitors are interacting with the rotor. You can see they have slightly different structures. This is an extended structure in the blue yeast inhibitor, and there's an alpha helical region in the bovine one, but otherwise they're pretty similar, but they bind at a different angle. And that has profound consequences, because actually the inhibitor, what we found by solving these 2 structures is that they've stopped the catalytic cycle at different points. And so this is a cross-section of the rotor looking upwards from the membrane. Here are the catalytic interfaces. There's where the nucleotide is bound. So that's one catalytic site. That's the second one, and that's the third. Now, if this were the bovine inhibitor, what would have happened is that this site, the empty or open site, would actually be empty. Whereas in the case of the yeast inhibitor complex, it's occupied by the nucleotide. And this is one of the structures that led us, but it has no associated magnesium. And this was one of the structures that led us to the conclusion that the nucleotide was released last. So we're now trying to exploit what we've observed by engineering inhibitor proteins to prop open this interface in different ways to try and trap different intermediates, with the aim of trapping a true transition state instead of using analogues and possibly other intermediates in this catalytic cycle.